The overall goal of this two-dimensional gel electrophoresis, coupled with mass spectrometry procedure, is to separate and identify human pituitary adenoma tissue proteome.
This method can help answer key questions in the proteomics field, such as high-throughput identity of proteome, larger-scale identity of low-abundance proteins, and discovery of protein speciation, protein 4, and protein species. The main advantage of this technique is that two-dimensional gel electrophoresis is easily coupled with high-sensitivity mass spectrometry to identify proteome, especially low-abundance proteins, protein speciation, protein 4, and protein species. The implications of this technique extend toward pituitary adenoma therapy because a key protein species or protein pattern is useful in patient diagnosis. Visual demonstration of this method is critical, as the two-dimensional gel electrophoresis steps are difficult to learn because it is time-consuming, labor-intensive, and involves several experimental tricks.
To begin this procedure, add 250 microliters of a previously prepared protein sample solution to each slot.